For our last section of Unit 4, we're going to get into how we make gametes, namely sperm and eggs. So this is the process, or meiosis is the name of the process, is used for sexual reproduction for organisms that require two individuals to create new offspring. So why do we care? One of the big reasons is that we sexually reproduce as humans, and it's really the reason why we don't all look the same, and you and your siblings don't necessarily look the same, with the exception of things like identical twins. This is how we are passing this different combination of genes on to individuals that determines who we are. And we'll get into genetics in the next unit, but this is kind of introducing us to that concept and helping us kind of smoothly roll into that next unit. So let's just kind of talk briefly about some of the ways that we can reproduce. There's really two main ways. And so if we are bacteria, or if we're a protist, a fungi, and some plants and some animals, they do reproduce asexually, meaning that one parent basically just makes a clone of itself, right? Something, an offspring that is genetically identical to the parent. But when we're looking at most animals and plants, we use sexual reproduction, where the offspring are genetically unique. So the odds of you coming up and having the combination of genes you have is, it's so rare. It's extremely rare. And the reason we're able to do this, the reason that the offspring are so unique, is because we are bringing two individuals together and bringing their gametes together. So you're getting half your chromosomes from one of those parents and the other half from the other. And so that process of bringing the gametes together, namely egg and sperm, we call fertilization. So let's go ahead and look at the kind of the process that we go through from adulthood to new offspring. So for sexual reproduction, and we're going to introduce some terms here, I'm going to try to get through them for you guys. We go through sexual reproduction. We have an adult female and an adult male, and both of these individuals have 23 pairs of chromosomes, giving each person a total of 46. Since they have their chromosomes are in pairs, meaning they have two sets, we call these cells diploid cells because there's two copies of every single chromosome. So there's 23 chromosomes, and in this adult female, for example, she will have two number 1s, two number 7s, two number 21s, two number 23s. And so two copies of every single chromosome. Now, since each individual has two copies, but we're bringing two individuals together to create offspring, we need to take those number of chromosomes and cut it down in half. So we go through the process of meiosis, and we'll get into those steps, where we take those pairs and we basically split them away from each other. So one set of chromosome ends up in the gamete. In this case, we're following the female, so it ends up in the egg, but if we were following the male, it would end up in the sperm. So 23 chromosomes total, one single set, so one copy of each. This is where we call the cells haploid. Think like you took the chromosomes, you cut the number of chromosomes in half, maybe that helps you. But haploid, we're talking about only one single set of chromosomes. Now, if we're making offspring, the egg and sperm, they will fuse together in a process we call fertilization, creating the first cell of a new organism, a new individual we call the zygote, right? So the fertilized egg is the zygote, and we're back to diploid because that one set of chromosomes came from the egg, one set of chromosomes came from the sperm. Everything's paired up again, right? So two number 1s, two number 8s, two number 16s, giving us 46 total chromosomes. And then just through mitosis, right, we're going to just make more of these cells through a bit of like a cloning process, right? Mitosis is somewhat like a cloning process. We create the actual offspring. And then if you watch this offspring grow into adulthood, we can circle back up here and start over again. So we're going from diploid to haploid and then back to diploid. It's a circle that we're making constantly. So because we are sexually reproducing, meaning we're bringing another individual into the mix, we have to take the number of chromosomes we have and cut it in half. That way we don't end up with extra chromosomes, right? Because we're fusing two cells together, they each have their set. If we had anything extra, we would end up with way too many chromosomes. So the process we're using to do that is called meiosis. So meiosis is going to produce those gametes, the eggs and the sperm, that are going to have half of that genetic information. Now, like I mentioned on the previous slide, if you have 2 copies of each chromosome, those cells are diploid. Think di is 2, right? If you remember back in the macromolecules lab and lecture, we talked about disaccharides, meaning 2 sugars. So diploid, 2 copies of chromosomes. And the gametes, the egg and sperm, they're going to have just 1 single copy of each chromosome, calling them haploid. Right? We bring those 2 we bring those 2 sperm and eggs together. We go through fertilization. We form the first new cell of an individual, the zygote, which is diploid now. Right. After that, we go through mitosis so that we can grow and develop and become an entire organism, freestanding on our own. We already went into mitosis, so I'm not going to review that here. Although if you are struggling with meiosis, you might want to revisit mitosis as well. What you're going to notice as we go through this is that there are a lot of similarities between mitosis and meiosis, and I'm going to kind of bring that up as we go through. However, while we're going through this, I'll point out the small differences, the important differences, but small nonetheless, that make mitosis and meiosis different. So meiosis has 2 main things that are going to happen. We're going to take the amount of genetic material we have and we're going to reduce it, and that we're going to produce non-identical cells in the end. Remember, mitosis was basically a cloning process. The parent cell is identical to the daughter cells, and the daughter cells are identical to each other. So nothing changes in the grand scheme of things with mitosis. But in meiosis, we want to create something that looks a little bit different from each other in terms of the combinations of alleles. We'll talk more about alleles in the next unit, but this is why we call meiosis a reduction division, because we're reducing how much DNA we have inside the cell. Now, when we're thinking about this, we're not just picking and choosing, we're not just randomly deciding which chromosomes we keep and which ones we throw out, right? So we're making sure that each daughter cell has the same genes, but that different versions of those genes end up in different combinations. And we'll talk a little bit more about that in just a second. So let's do a quick little overview where we're looking at meiosis, where we're reducing the genome by half. And so what we're doing there, we have interphase, just like we saw with mitosis, where the DNA gets replicated and all that happens. Now we're color coding it with maternal chromosome being in this darker green and the paternal one being in this lime green. Now, because we're in pairs, we have some those pairs of chromosomes we call the homologs or homologous sisters or homologous pairs. My apologies. Homologous pairs. So homologous pairs have the same type of genetic information on them. So let's say we're talking about eye color. Both of these chromosomes will have the gene for eye color, but the maternal one might have the brown eye allele, so the brown eye version of that gene, and the paternal side might have the blue eye allele or the blue eye version of that gene. So same type of information, just not exactly identical. And so the homologous pairs are similar, same kind of size, same basic shape, same basic structure, but they do differ slightly in terms of the versions of each gene we find there. When we go through meiosis, we're going to make sure that the pairs split away from each other. That's our main goal. And then for meiosis 2, we're going to split the sister chromatids. So honestly, meiosis 2 looks a lot like mitosis. So if you, again, make sure you know mitosis. It's going to help you understand meiosis. It really will. Now, with meiosis, we are only producing sex cells called gametes. We learned in mitosis we were making skin cells, liver cells, cells found in the lungs and the digestive system, right? All basically, if you think of a cell, you're probably thinking of one that used mitosis. But in meiosis, we're only producing gametes. That's going to be the egg and sperm. And so when we're producing those, since we're having the number of chromosomes, we're creating just one set of 23 from each parent, right? Since we're getting those 23, when we bring the two sets together, we're going to have, again, 2 number 1s, 2 number 5s, 2 number 13s, so on and so forth. The ones like the 2 number 1s that go together are called homologous or homologs, right? So the homologous chromosomes, again, like I said, they have the same type of information. They're the same size, the same length. Like, for example, they might have they're both going to have the eye color gene, but they might have different versions of those same genes, like blue versus brown eyes. Within that set of 23, we have the last one. So pair number 23 is going to be the sex chromosomes. This is going to determine male from female. And so we have X's and Y's. Females will be XX, males will be XY. Now, do remember, before we go through meiosis, just like we saw in mitosis, we are going to replicate all of those chromosomes, right? If we don't replicate them, we're not going to have enough DNA in order to go through meiosis, the whole process. So we have to replicate it before we even get started. So here are the homologs and the sister chromatids. So these two here, we have a maternal chromosome and a paternal chromosome. So this chromosome came from this person's mom, and this chromosome came from this person's dad. The homologs are extremely similar, but not identical. That's a big thing. Similar, but not identical. Same type of information, but different versions. Think of it like candy brands. The gene would be like a candy brand, like Lifesavers. But the maternal chromosome might have the version of Lifesavers that gives you cherry, and the paternal might have a Lifesaver version that gives you pineapple. They're both Lifesavers, but different flavors of the same candy. So these are the homologs. We go through replication, so we see this nice little X-shaped. And there we have the sister chromatids. So if you remember sister chromatids from mitosis, you know that the sister chromatids are identical. So these two pieces I'm pointing to are identical to each other. These two pieces I'm pointing to over here are identical to each other, but the dark green and the light green are not identical to each other. So dark green and dark green are sister chromatids. Light green and light green are sister chromatids with each other, but dark green and light green are homologs. So when we're going through meiosis, we're looking at 2 cell divisions compared to mitosis, which only has 1, right? We're going through 2 of them because, again, we're trying to cut the number of chromosomes in half. Both of these processes are going to have interphase, but one of the big differences, one of the big changes we're seeing between meiosis and mitosis is in the very beginning of meiosis, in prophase 1, we have a process called crossing over. Crossing over is where those homologs get really close together and they can actually trade some pieces of information from one chromosome to the other, giving us what we call genetic recombination. This is why we end up so diverse and different from each other, and why you can look at your siblings and see the similarities, but you don't feel like you're a twin or a clone of your siblings. So this basically gives us different combinations of the versions of genes that we have on our chromosomes. So crossing over, right, here is the mom side of things. Here's the dad side of things. These are homologs. They're going to get really close together, and they get so close that they can actually trade pieces. Now, if we notice, we're trading the same sections. So like, we're not going to trade this upper arm with the lower arm. We're trading this lower arm at about the same level. So this way we make sure that every single chromosome still has the same type of information. So the way I like to explain it is thinking about like cupcakes and the lunchroom when you were a kid. So think about if your mom packed you a cupcake in your lunch and someone came up to you and said, hey, I'll trade you your chocolate cupcake for my celery sticks. You'd probably look at that person like they were crazy, because who's going to trade away their wonderful chocolate cupcake for celery? You're probably not going to do that. But if someone came up and wanted to trade your chocolate cupcake for a vanilla one, you might be more on board to trade because it's an even trade. No matter what, whether you decide to trade or not, you still have a cupcake. And so that's kind of what we're doing here. We're making sure that we're trading the same sections of the chromosomes so that we end up with the same type of information. And so what we see is after we go through this process, we now have 4 chromatids, but none of them are identical anymore. Remember, we started with the sister chromatids being identical, and now we have all green or all dark green with some light green with some dark green, and all light green. So everyone's a little bit different. This is, again, why you end up looking different compared to your siblings.